In this module, I will discuss about transposable genetic elements in bacteria. DNA sequences that can move from one position of the genome to another is called transposable genetic element or simply transposition. Types of transposition. Transposition are classified into three types: insertion sequences or IS elements, or it is also called cut and paste transposition, composite transposition, and TN3 elements or replicative transposition. Structure of IS50 element. This is the one of the most simplest transcription. It is bordered by terminal inverted repeats of 9 to 40 nucleotides. Then IS50, when it is inserted in the target site, there is a target site duplication occur, which is shown in the figure. Composite transposition. Composite transposition is designated by TN. It is designated by TN. In the composite transposition, there are two IS1 elements, each one of 768 nucleotide pairs. There are 23 nucleotide pair inverted terminal repeats, and two IS1 elements binds the chloramphenicol resistant genes. So CAMR gene is flanked by two IS1 elements. So this one is called composite transposition. Now comes the mechanism of transposition, that is how transposition occurs. IS elements usually encode a protein which is called transposons, that is needed for transposition or transposition of genetic element from one position to another. The transposes binds at or near the ends of the element and then cuts both the strands of DNA. IS element is inserted and target site duplication occur. So in this figure we have described, that is the how insertion occurs and what is the mechanism of transposition. So target DNA is clipped by transposes as a result of the staggered cleavage, which is shown over here. A space is created, an IS element is inserted in this space. This IS element is cut by a transposes enzyme and it is inserted in the space created due to this staggered cleavage. DNA synthesis then fills the gap. So here there is a target site duplication occurs and transposition takes place. So this is the mechanism by which transposition occurs. This is also called cut and paste type of transposition. Next comes genetic organization of TN3 elements. Now TN3 elements consist of three genes, that is TNPA, which synthesize transposes, TNPR, which synthesize resolvase or repressor, and BLA gene, which synthesize beta-lactamase. This beta-lactamase, which gives resistance to ampicillin, and there are 38 nucleotide pair inverted repeats are there at two ends. Now the transposition of TN3 elements. There are two types of plasmids are there. It is called, one is donor plasmid which carries TN3 elements, and there is a recipient plasmid which is devoid of any TN3 elements. Now the transposes, which is synthesized by TNPA gene, made the co-integrate. A co-integrate is formed with the help of transposes, where replication of TN3 element takes place. After formation of co-integrate, TN pair which synthesize resolvase. This resolvase catalyze the process of recombination. After recombination, it resolved to a donor plasmid and a recipient plasmid, and each of them carrying a TN3 elements. So there is a transposition of TN3 by the formation of co-integrate. It is also called replicative transposition. Next comes, what is the medical significance of transposition? The medical significance of transposition are that is resistance gene, it spreads to other bacterial population. Here in these illustrations we are showing that is in course of evolution, that is conjugative plasmids or resistance transfer factors are originated, and there are conjugative plasmids which facilitate conjugations and it passes during the formation of conjugation tube. It undergoes recombination with non-conjugative R plasmid which carries a streptomycin resistance gene, and also it carries an IS1 elements. Now conjugative plasmid which carries RTF factor, that is resistance transfer factor, that is it facilitates the transfer of resistance factor. Now in the IS1 elements which is present both in non-conjugative R plasmid and conjugative R plasmid, there is a recombination occurs and a larger R plasmid forms. It is called conjugative R plasmid. This conjugative R plasmid has RTF component or resistance transfer factor components, two IS1 elements and streptomycin resistance gene. This is called R determinant, and so it has got main two components, R determinant, resistance determinant, and RTF components. So this conjugative R plasmid transfer the resistance gene horizontally to different bacterial populations, as a result of which resistance genes by means of transpositions it spreads to the bacterial populations and resistance to antibiotics develops. So this is the one of the most important medical significance of transposition. So Barbara McClintock, she is responsible for first discovering the transposition, the mechanism of transposition in maize. For that contribution in genetics, she was awarded Nobel Prize. In this module, we have provided key concepts and summary, list of books for reading, some quizzes are there, and study materials. Moreover, if you have any question, please feel free to ask us. Thank you for participation.